Welcome back to the playlist on lactate dehydrogenase and glycolysis. In this video, we're going to go over the mechanism of an enzyme known as L-Lactate dehydrogenase. And eventually in this video, we're going to do the organic mechanism of this enzyme, but like all videos that are going to be in this playlist that are new, I want to give you some basic background information on this enzyme and why you should think it's important. Okay? Now, the process that lactate dehydrogenase is involved in is something that's known as anaerobic fermentation. And in Homo sapiens, in humans and other mammals, the molecule that's involved in fermentation is known as L-lactate, specifically the L isomer. Okay? And to understand why this is, we need to look at this pathway that's shown over here, known as glycolysis. Now, you can get glucose through the diet, certainly, and there's also ways that the liver has to biosynthesize glucose, which is a process known as gluconeogenesis. But in any case, when you're a cell that needs energy, glucose is certainly a good candidate. So through glycolysis, the cell will catabolize glucose into two molecules of pyruvate. Glucose is a 6-carbon sugar, so this molecule right here, this is 6 carbons. And if you catabolize it down to 2 pyruvates, these are 3 carbon molecules each. Now, but to understand why lactate dehydrogenase is important, I need to make an assumption, okay? I need to make an assumption that I'm under anaerobic conditions, okay? Because this is the condition under which lactate dehydrogenase becomes exceedingly important. So I'm under anaerobic conditions, and what exactly does that imply? Well, anaerobic conditions implies that I have low molecular oxygen. Okay, that's something very important. If I'm under anaerobic conditions, that implies that my level of oxygen that I have is greatly diminished. Okay, I still have it, but there's just not near as much as there would be if I was running aerobically. So anaerobic just implies there's a smaller amount of oxygen present. Okay? Well, why is that important? Well, what I want to have you bear in mind is that through glycolysis, we're going to generate a very critical reduced coenzyme. And I'll circle it in green. This coenzyme is referred to as NADH. The abbreviation stands for nicotinamide adenine dinucleotide hydride, the reduced form of NAD. Now, the reason that NADH is critical is because we're going to have this NADH, okay, and through a mechanism that we're not going to elucidate in this video, the NADH effectively gets transported into the mitochondria. So one thing that's important to realize is the glycolytic enzymes, along with lactate dehydrogenase, are cytosolic, but the NADH that's made in glycolysis is essentially transported into the mitochondrion, okay? Now, why exactly is that important to realize? Well, keep in mind, we're making the assumption that in this condition, we're running anaerobically. Okay, so 2 things. Number 1, this enzyme that's shown right here, this was what we often refer to as complex 4, and if you really want to get complicated as to what the name is, it's called cytochrome. This is called cytochrome C oxidase. And one thing that you should be aware of with respect to this enzyme is its substrate is molecular oxygen. Okay, so just keep in mind we're running on low pressures of oxygen. Okay. Also, I have this other enzyme in here, which is referred to often as complex 1, although sometimes you'll hear it referred to as NADH dehydrogenase. And what the purpose of NADH dehydrogenase is it's to take this NADH that we made in glycolysis and other processes like the TCA cycle, and it's going to reoxidize NADH back into NAD, okay, into NAD as shown right here. And then if we had lots of oxygen present, this NAD could then go and react in another cycle of glycolysis, okay? At least of the enzyme glyceraldehyde 3-phosphate dehydrogenase. But here is the problem, okay? If we're running in conditions when we have low oxygen present, where we can't get oxygen as readily to the tissues, this process of cytochrome C oxidase shuts down. at least to a very large extent. So cytochrome C oxidase can no longer catalyze very much of its reaction because we have such low oxygen, and oxygen is a substrate for cytochrome C oxidase. I hope it makes sense to you that if an enzyme doesn't have its substrate, its turnover goes greatly down. Okay? So notice what cytochrome C oxidase is doing. It's pumping protons into the inner membrane space of the mitochondria. That's part of the mechanism of cytochrome C oxidase. And notice also that this NADH dehydrogenase that reoxidizes NADH back to NAD, it also pumps protons from the matrix into the inner membrane space of the mitochondria. So all of these protons being pumped into the inner membrane space, what is the function of doing this? Well, it turns out that there's another enzyme that's next to the respiratory chain, or the electron transport chain in the mitochondria, known as ATP synthase. Okay? And the function of ATP synthase is to use the pumping of protons to power the synthesis of adenosine triphosphate. So these protons that were originally pumped in by complexes 1, 4, and then also there's complex 3, which we didn't mention, those protons will essentially rush back through a subunit of ATP synthase from the inner membrane space back into the matrix. Okay? And that movement of protons creates mechanical energy that powers ATP synthase. Now, what's important about this? Well, whenever you use the respiratory chain for your metabolism, that's normally what you consider aerobic respiration. Okay? And if you're doing aerobic respiration, a general estimate as to how much ATP you produce per glucose is generally between 30 and 36 ATP. That is quite a bit when you come back and you compare this to glycolysis, where we essentially in glycolysis get only 2 net ATP per glycolytic reaction. Okay? Because keep in mind we burn 2 ATP with hexokinase and phosphofructokinase 1, and then we gain 4 more through phosphoglycerate kinase and pyruvate kinase, so the net is 2. Now, a question you should ask yourself is would a cell be happier running on 2 ATP or about 30 to 36 ATP from the electron transport chain? And the answer is obviously the cell would much rather run on the 36 ATP from the respiratory chain. But here is the overarching problem. If you have low oxygen that's getting to the tissues, cytochrome C oxidase complex 4 has trouble running because it doesn't have its substrate. And if cytochrome C oxidase shuts down, then all of these other enzymes back here also shut down. Okay? And if they shut down, that means that this process right here of going from reoxidizing NADH back to NAD, this does not happen. Okay? If cytochrome C oxidase can't catalyze its reaction on oxygen, the processes before this in the respiratory chain, i.e. complexes 1, 2, and 3, they essentially shut down. Okay? And you don't get regeneration of NAD. Well, the question is why is NAD important? Well, NAD is mainly important from a general physiological perspective in 2 ways. Number 1, notice that NAD is a substrate for glyceraldehyde 3-phosphate dehydrogenase. So if you're not regenerating NAD, this enzyme shuts down. Okay? And then you also have to bear in mind that there's also several TCA cycle enzymes that utilize NAD. So if you're not regenerating NAD, those also shut down. So regenerating NAD is extremely important for maintenance of ATP. And the way that it works is NADH dehydrogenase reoxidizes the NADH generated through glycolysis in the TCA cycle back into NAD, and then the NAD can be reused in other enzymes. Okay? And the way that NADH dehydrogenase functions is only when cytochrome C oxidase is running. But cytochrome C oxidase doesn't run very well when there's low oxygen. So that means that if you're running anaerobically, i.e. you have very low oxygen present, the amount of ATP that you produce is incredibly low. It's basically reduced to the ATP produced by glycolysis, which is about 2 per glucose. Okay? Now, What's important to realize is cells do not like to run on purely glycolysis. Very few cells ever want to run on that, especially cells like the heart. They're so dependent on running on the electron transport chain. So your cell has a way to get around not reoxidizing NADH back to NAD, which was the function of NADH dehydrogenase. So the way that the cell gets around reoxidizing NADH back to NAD is by using an enzyme known as lactate dehydrogenase. Okay? So this pyruvate right here, this 3-carbon fragment of glucose, this pyruvate will not go on to react in the pyruvate dehydrogenase complex because that enzyme also requires NAD. Instead, what will happen is the cell will effectively use lactate dehydrogenase and it will waste an NADH and reduce the carbonyl, at least the ketone of pyruvate right here. It will reduce this ketone in a carbonyl reduction to a hydroxyl group which is present on lactate. Okay, so we'll go over the mechanism of that shortly, but I want to make sure you understand this. What the cell does is it uses lactate dehydrogenase and it essentially reoxidizes NADH back into NAD by wasting the electrons, i.e. the hydride, on the carbonyl of pyruvate, which generates lactate. Now, lactate doesn't really do anything in humans. It's a waste product. And it signals that if this continues, the cell is going to die. So eventually you'll have mechanisms to reoxidize lactate back into pyruvate, and we'll discuss that in another video. But I want to make sure that you understand what the function of lactate dehydrogenase is. If NADH dehydrogenase in the respiratory chain shuts down because of low oxygen, you have to have a way to regenerate NAD+, and that's by wasting the hydride, the electrons, on pyruvate, reducing it to lactate. Okay? And so that effectively regenerates NAD+, and so that NAD+ can then be used in another cycle of glyceraldehyde 3-phosphate dehydrogenase and other enzymes. Okay, so that's a little bit of the background information on lactate dehydrogenase and the bare minimum that you need to understand how it works. But now what I want to do is I want to go over the organic mechanism of lactate dehydrogenase. And the very first thing I'm going to do is I'm going to show you the conversion from pyruvate to L-lactate. So this is the direction we're running first. Okay, and so what ends up happening is whenever you have lots of NADH around and you're not regenerating any of the NAD+, this is the direction that the reaction will run. So in other words, if I come back down here, okay, if I'm running when there is low oxygen, in other words, I'm running anaerobically and doing anaerobic fermentation, that means I'll be running this direction. So this direction represents low oxygen. I'm running anaerobically and doing fermentation. Okay, so some things about the lactate dehydrogenase or LDH active site. Okay, number one, there's a binding site for NADH. So whenever you're running in the direction from pyruvate to lactate, you're going to have to somehow bind this nicotinamide adenine dinucleotide hydride in there. And of course, I've grossly abbreviated the structure because it's very large and I don't want to have to draw it every single time. Okay, there's also a special binding site for pyruvate. So that's what this molecule is right here. This is our pyruvate molecule, and the pyruvate is stabilized in here through several residues. Number one, notice that the carboxylate, it's in the deprotonated state in terms of the enzyme's mechanism, and the negative charge that exists on the carboxylate is stabilized in the active site by a critical arginine residue that's up here. So this arginine residue is serving as an electrostatic donor to the carboxylate of pyruvate. So this guanidino group of the arginine has a positive charge overall, and so it's able to electrostatically stabilize the carboxylate of the pyruvate. Also, if you look at this carbonyl right here of pyruvate, you'll notice that the oxygen has a partial negative charge. And that partial negative charge is also stabilized by another arginine residue that's in the active site. So again, the arginine has a guanidino group with a positive charge, and that's able to stabilize that carbonyl's partial negative. And also, a critical histidine residue in the active site as well stabilizes that carbonyl's partial negative charge. And very similarly to what we saw in trypsin's mechanism, we have a histidine residue in here that's going to act as a Bronsted-Lowry acid base, or we could call it general acid base residue. And we also have an aspartate residue. The carboxylate of the aspartate is hydrogen bound to this particular hydrogen of the histidine residue, and that's what stabilizes the histidine in the correct position to allow the proton transfer to occur. So hopefully all of that stuff makes sense. Now let's actually look at the mechanism and how it works. I will do the mechanistic steps in green. So hopefully you have a little bit of intuition on how NADH physiology works. So one thing that's important to realize about NADH is that when it's doing this reduction, it likes to regenerate aromaticity of the pyridine ring. Okay, so what happens is this lone pair kicks in here to form a pi bond there, and that forces this double bond to come right here and essentially eject a hydride as a nucleophile. And the hydride, just like you saw in organic chemistry when you did reductions with like sodium borohydride or lithium aluminum hydride, it's exactly the same thing, except the hydride donor here is NADH. So what ends up happening is the hydride comes out and nucleophilically attacks this carbonyl carbon right here. Now what essentially happens from there is the carbonyl bond splits, the pi electrons come out, and they abstract this proton from the critical histidine residue in the active site. And so what you end up getting at that mechanistic step is you get this molecule, which is called L-Lactate. And keep in mind, it's still in the deprotonated state. And partially what happens is the electrostatic interaction right here between the carboxylate and this critical arginine residue occurs. And then what's going to happen is you have to regenerate the resting state of the enzyme. Because notice the way that I started was with this critical histidine residue in the protonated state. So what's essentially going to happen is a hydronium is going to be allowed into the active site, and the critical histidine residue will deprotonate the hydronium, and that essentially regenerates the histidine, still electrostatically hydrogen bound, to this critical aspartate residue in the active site, and you get lactate dissociation, and then you get NAD plus dissociation. Okay? So hopefully that mechanism made a little bit of sense. But going back to what we were talking about earlier is, you have this emergency sort of biochemical valve right here, where if you start building up lots of NADH and you can't reoxidize it back to NAD, you use this reaction. So this occurs basically under anaerobic conditions. But what happens when I go back up in partial pressure of oxygen? So in other words, if I build up my oxygen levels again, so I'm able to get oxygen to my tissues, then I'm going to be operating under aerobic conditions, in which case I am able to reoxidize NADH back to NAD. Well, what's going to happen then, generally, is you're going to reverse this reaction. So the lactate that you formed through reducing pyruvate is going to be reoxidized back to pyruvate. Okay, and that's the next mechanism that I'm going to show you. So this mechanism that I'm showing you now, this is essentially the mechanism that occurs when you are running from lactate. This is from lactate to pyruvate. And this situation occurs whenever you go back into aerobic conditions. So when you go into aerobic conditions and you're able to get oxygen to your tissues at a good rate, you're able to do this reaction where you reoxidize lactate back into pyruvate. And then the pyruvate can, of course, go into the pyruvate dehydrogenase complex, and the acetyl-CoA generated there can then go into the TCA cycle. Okay, so what you're assuming in this mechanism here, which I'll do the steps in green again, is you're assuming that this critical histidine residue that we talked about is in the deprotonated state. And if it's not, you can always do a proton exchange with solution. So what's going to happen is you're going to get a carbonyl forming reaction. Okay, in the reverse reaction, what we did earlier, this was called a carbonyl hydride addition. When you do a carbonyl hydride addition, you get a hydroxyl group. But we start off with a hydroxyl group here, so now we're doing a carbonyl formation reaction. So what's essentially going to happen is this critical histidine residue is going to deprotonate the hydroxyl group of lactate. And that's going to induce these electrons right here to come in and form the carbonyl. Now, this carbon right here, if nothing else happened, you'd have 5 bonds. because there would be 4 sigma bonds and 1 pi bond, and that certainly cannot be the case. So what ends up happening is you have loss of a leaving group, and it turns out that the leaving group is this hydride right here. So what ends up happening is the hydride is ejected, and it attacks this carbon atop the NAD ring. Okay, and that's going to essentially force a double bond rearrangement in which this nitrogen right here acts as an electron sink, and these pi electrons end up on top of the NAD nitrogen, and then you end up with reduced NADH. And notice the other product I get when I do carbonyl formation is pyruvate. Okay, and I regenerate the resting state histidine. I have all my arginine residues in here that are electrostatically stabilizing things, and then the aspartate is electrostatically hydrogen bonding with the histidine residue, providing proper orientation for the general acid-base catalysis. Okay, so hopefully that made sense. Let's do a very quick recap of the mechanism. So if I go from pyruvate to lactate, notice that what I'm getting is hydride ejection from NADH. So NADH is acting as the reducing agent, in which case it's getting oxidized into its NAD plus form, and I'm doing a carbonyl hydride addition, so I'm going to get a hydroxyl group. So the aromaticity reforms in this pyridine ring, and the hydride gets ejected, and it essentially attacks this carbonyl carbon of pyruvate, and that's going to cause these pi electrons to break out and abstract the proton from this critical histidine residue stabilized by the aspartate. And in that one mechanistic step, I generate lactate. So here's the lactate, and it will eventually dissociate from the enzyme along with the NAD, and then a proton exchange with a hydronium from solution is going to regenerate the resting state of the critical histidine residue in the active site. Now, if I'm running under aerobic conditions and I want to get rid of the lactate and regenerate pyruvate from it, I can essentially reoxidize the lactate into pyruvate by essentially reducing the NAD plus into NADH. And the way I'm going to do that is by doing general acid-base catalysis and doing a carbonyl formation reaction. So this critical histidine residue deprotonates the hydroxyl group of lactate, and that induces carbonyl formation to form pyruvate and ejection of this hydride right here to attack the carbon atop the NAD ring, and then the nitrogen right here of the pyridine ring acts as an electron sink, and you get formation of reduced NADH and pyruvate. And that's essentially the mechanism of lactate dehydrogenase. In other videos, we'll go over some more facets of this enzyme that are rather interesting. So hopefully that video gave you a little bit of intuition on the mechanism of lactate dehydrogenase. See you in the next video.